Hello everyone, my name is Caleb Tong and currently I'm in my final year of chemical and biomedical engineering. This talk will be covering the topic of categorization of structure-based constitutive models for aortic wall tissue under planar biaxial tension. A little bit of introduction. Aortic aneurysm is the enlargement of certain regions of the aorta, the largest artery in your body, due to microstructural changes in the aortic wall tissue. It is estimated that complications from aortic aneurysm, such as dissections or ruptures, can lead to 97 to 100% total mortality rate with the surgery. The number decreases to 5% for cases with elective resection. This stresses the importance of developing accurate diagnosing tools to predict when surgery is needed. While current criterion for surgical intervention is based on the diameter of the aneurysm, aortic wall tissue's mechanical behavior also plays an important role. To test out the mechanical behavior of the aortic wall tissue, we put a physical piece of the aorta on the biaxial mechanical testing when it is stretched in both directions, provide relationship between stresses and deformation. Because the physical sample is obtained post-surgery, the process is very invasive, and this is where modeling comes in. Structure-based constitutive models are mathematical models that help to capture the mechanical behavior of the aortic wall tissue while correlating back to the histological and microscopic data. This research focuses on the Hosafeld 2005 model. The objective of this research is to understand the incorporation of useful data to improve the prediction capabilities of structure-based constitutive models on a new experimental data set. And by useful data, what I meant is the MRI imaging and histological results. Before moving further on, two important definitions that you need to know: Washer stress and elastin fragmentation. Each heartbeat produces pulsatile blood pressure and have forces generation within the wall. Acting forces per area of the arterial wall is the Washer stress. Elastin is one of the important fiber or protein that bears the mechanical load, and under the effect of Washer stress, elastin can be discontinued or fragmented, leading to microstructural changes and eventually wall weakening. Methodology. Biaxial data results with clinical data as shown are split into two groups: descriptive group, which is 70% of the total number of samples of data, and the predictive group, which is the other 30%. The descriptive group data, with the support of several software such as Hyperfit, Python, and MATLAB, are used to generate the average material mechanical behavior model, and this model is then applied back to the predictive group to see how well it can fit the data. The metrics measured here for comparison purposes is the R-square, and the process is repeated 3 times to increase number of data points and overcome limitations of number of samples. After that, certain samples are selected based on either normal or high Washer stress, elastin fragmentation 0 and 1 graded to generate the average material curve. We're interested to see by grouping samples rather than compiling all of the data can actually help to improve the predictability of the average material mechanical behavior model. And the result is actually this. From left to right are the values of R-square from no categorization, categorizing them into normal and high Washer stress, elastin fragmentation 0 and 1 graded respectively. All yellow highlighted p-values shows that the difference is statistically significant except for the case of high Washer stress. In conclusion, constitutive model plays an important role in the pipeline of developing a new, more accurate diagnosing tool. But taking a step back and finding the missing link is also important. In this research, by combining engineering and medicine approaches, specifically incorporation of Washer stress and elastin fragmentation data, we can improve the predictability of constitutive models on a new experimental data set. Future directions, which is also what I'm working on right now for my final year thesis, is to understand better the effect of extracellular matrix in the arterial walls, specifically the collagen fiber contains an orientation of the aortic wall tissue's mechanics. This will provide more insights toward the pathology and development of aortic aneurysm. Thank you for listening.